I'd like to thank the organizers at LDRTC for inviting me to give this presentation in this time of the COVID virus, and the organization of getting this done virtually is really an undertaking. I'd like to thank Dr. Unismaz for her presentation, and I'd like to follow on with that with some additional information. I am Gregory Grabowski. I'm a professor emeritus in the departments of pediatrics and molecular genetics and biochemistry at the University of Cincinnati College of Medicine and the Children's Hospital Medical Center and the Division of Human Genetics. What I'm going to speak to today is some of the relevant mechanisms of the SARS-CoV-2 infections and the potential implications for lysosomal storage diseases, LSDs. Some of this is speculative because the entire mechanisms of the COVID virus 2 are not known. A lot of this presentation will be based on previous SARS-CoV investigations, and I'll take you through this. Oh. These are my disclosures. There's basically nothing relevant here to this presentation except for Kiniksa Pharmaceuticals, which I will mention something about a new paper that just came out toward the end of the presentation. It just came out yesterday, as a matter of fact, June 17th. What we're planning to do is have all the participants at the conclusion of this activity to be able to define and differentiate between lysosomal storage diseases, inflammatory mechanisms, and those associated with COVID-19 or other coronaviruses. To define a plan for clinical care, monitoring of LSD patients with COVID-19, and to read and be able to interpret relationships of new COVID-19 inflammation data relative to lysosomal storage disease, immune dysfunction. In the lysosomal storage diseases, the inflammatory findings differ. And the pro-inflammatory effects appear to be more profound in the glycosphingolipidosis, GSLs, but are present in most of the lysosomal storage diseases. The cell type of involvement impacts the nature of the inflammatory response. For example, macrophages versus neurons versus endothelial cells. And these are responses to basic disease processes, but are incompletely defined. The mechanisms are more fully understood in Gaucher disease, particularly the visceral disease, and this will be the focus of most of this presentation, with some indications of how this applies to the other lysosomal diseases. At first, I want to speak to the initiation and propagation mechanisms of Gaucher disease in COVID-19. They will be in that order, and I'll show you how this all fits together as we move through. So initiation and propagation are important because they're different. The disease state is relatively simple and causally driven at the beginning of the disease. So that's the initiation. In Gaucher disease and other LSDs, it's the genetic defect and the enzyme abnormalities. The cells and tissues can tolerate specific deviations from normalcy, and things can be fairly calm and moving forward in a slowly progressive or more rapidly progressive nature. But the evolution of the cell and tissue-specific temporal involvement has increasing complexity with it. And there's a progression from pro-inflammatory dysregulation that leads to changes in cell type and the nature of cell death. You're going to see this figure several times during this presentation. And this is an overall view of the disruption that occurs within cells during lysosomal diseases, and I'll show you how later this fits in with COVID-19. A master regulator of all of this is mTOR complex 1, or as I like to call it, mTORCH1. And this sits on the lysosomal membrane when everything is normal and there's a normal nutritional intake and there's no stress on the cells. And when it sits on this lysosomal membrane, it is activated and can phosphorylate two major transcription factors of lysosomal function, TFEB and TFE3. When they're phosphorylated, they cannot enter the nucleus, and therefore they're inactive. But during times of cellular stress, mTORCH1 leaves the membrane and becomes inactive, and therefore does not phosphorylate TFEB and TFE3. They enter the nucleus and turn on an entire transcriptional program for autosomal function, lysosomal function, peroxisomal function, and I haven't shown it here, but mitochondrial function also. As you can see, this is a very complex metabolic pathway and can lead to disruption of any of the parts of the pathway, including the autophagy pathway and the lysosomes, which can become disrupted, have poor protein function, and indeed have altered integrity. And this can occur from either the substrate increases or a decrease in a precursor or a direct activation by some other mechanism. So this is a very complex system, and it leads to, as the disease progresses, another component to this. Here's what we just saw, this entire mechanism over here. And during time of starvation and stress, this is much more than just storage and lysosomal autosomal dysfunction. It's also inflammatory dysfunction depending upon the cell type. Here I'm showing what would happen, for example, in a macrophage, where you hit this entire disruption of the system and activation to make more lysosomes, but they're dysfunctional. And because of these effects of substrate or decreased precursor or direct activation, you get activation of NF-kappa B, you get pro-IL-1 beta and pro-IL-18, and activation of the inflammasome. Once that happens, you're on a pathway to cell death. And it can either be pyroptotic with massive fever and inflammatory response, or it could be apoptosis. Also, a compound by the name of RIPK1 or 3 can participate in this process and lead to necroptosis. Now, what happens with necroptosis or pyroptosis? These are major inflammatory effects in the cell, and they draw in other cells to create a massive inflammatory response against a disease process that is happening because the cells are dead and they're sending out eat me signals. In Gaucher disease, what you see here is that in lungs, for example, in the mouse model here, you see increases in macrophages, dendritic cells, polymorphonuclear leukocytes, T cells, liver, in the spleen, liver, and lung. And what happens is that these storage cells that we call Gaucher cells, full of glucoceramide, start spitting out chemokines and cytokines to draw in other cells of different cell types to affect and a control over the inflammatory response. And importantly, something we'll come back to is the type 1 interferon, which has the mechanism of downregulating this massive inflammatory response. So untreated, this is what happens in Gaucher disease. It causes a terrific metabolic storm, and I think you've heard that before. But we've done work, and others too, where we have connected this massive cellular response and inflammation response from the innate immune system into the adaptive immune system. And what has happened here is in Gaucher disease, the body recognizes somehow by breaking tolerance, recognizes glucoceramide, the storage substance, as an antigen and makes antibodies to this and creates an antigen-antibody complex and an autoimmune disease. This then activates the complement system, and in this case, C5a, which is massively increased. It hooks onto its receptor, and it signals, surprisingly, an increase in glucoceramide synthase, increasing the storage substance that is already causing the disease and causing a vicious cycle. Similar kind of thing we'll talk about in COVID-19, where you get this amplification or a vicious cycle of the disease, leading to a massive pro-inflammatory response. So let's move to COVID-19. Once we have that basic sort of cellular mechanism set forth for an inflammatory response due to this lysosomal disease, what about a viral disease? And as you've all heard on the news, there are at least two forms of COVID disease. There's a mild and moderate form, and there's a very severe form. And in a recent publication, they put out these nice graphs that show us approximately what's happening. And here in red, we have a viral load that increases and then decreases. And there is a type 1 interferon response that is very high. In addition, you get kick-in of lymphocytes and production of antibody to the virus and slowly dampen the inflammatory cytokine process. In the severe disease, things get out of hand. The viral load is larger, the interferon type 1 response is lower, and therefore viral replication can occur, because the goal of type 1 interferon is to dampen viral replication. This leads to a massive increase in disease severity parallel with the inflammatory cytokine productions. Now, I'll introduce you to this virus. This is a very interesting biological compound, I have to say. This is the whole genome. The entire genome is 30 kilobases long. And of course, relative to humans, where we have 3 billion bases long, our genome, this is a very simple organism. And I'll show you more about this genome structure in a minute. But there are an S, an E, an M, and an N protein, which are the spike protein, the matrix protein, the nuclear capsid, and the envelope protein. Those are structural. The rest of these genes code for non-structural proteins and have different functions. Here's a bigger view of this genome and what it does. And this is not, it just doesn't go in and make itself. The importance of these origin of replications, 1A and B, is that they make replicates of the genome so that the virus can then move on to infect other cells and make more of itself. But you'll notice all these other little non-structural proteins that are here have different functions. 3B becomes very important because it controls the release of cytokines and chemokines and the inflammatory response. 7A regulates NF-kappa B and starts the entire inflammatory process. 8A participates in apoptosis and necroptosis. 3A causes direct lysosomal damage and necroptosis. 6 limits the production of interferon 1, therefore promoting the development of more viruses. 7B has nebulous functions, but are complexing with other proteins that are here. 8B attaches directly to the inflammasome protein, increasing the activation, and 9B functions by dampening interferon production and leading to apoptosis. Here we see an overall view of what happens during the coronavirus infection in cells. The virus comes from outside the cell, attaches to its receptor, the ACE2 receptor, enters the cell, and then gets trafficked to the lysosome, where it is modified and its RNA is released and produces RNA for translation. And once it does that, it starts producing all these proteins. RIP3 and 3A form a complex which disrupts the lysosomal function and dissociates mTORC1 from the lysosomal membrane, causing an upregulation of TFEV and TFE3 to make more lysosomes and cause a production of a beginning of a storm for cytokines. 8B causes ER stress, therefore amplifying the effects on inflammation. It also disrupts lysosomal function, and it binds directly to the inflammasome. 6 downregulates type 1 interferon, leading to an inability of the cell to shut off viral replication. This all leads to this very large inflammatory response with these initial cytokines and chemokines, leading to necroptosis and pyroptosis. And what I said before was these now are dead cells. And these cells are saying, come get me, eat me, call in more cells, more inflammatory response. And this is nicely put together in a recent paper that is a very complex system here that you can see. The point I want to make out of this, because we've talked about most of this before, is this leads to a cytokine storm. And in Gaucher disease, you don't have this downregulation of type 1 interferon. That could be very important. So if that's how you get the cytokine storm and the respiratory disease syndrome and the amplification of this viral disease. One thing that's unclear among many is the neurological involvement in this disease. And I want to focus just a little bit on this thing called septic encephalopathy delirium. And what this is the following. This is part of a system of an effect on microglial cells in the brain. Since we do not know if the COVID-19 virus actually infects the brain, this system we do know does affect the brain during a cytokine storm. The microglial cell is primed by previous exposure to viruses and other things, and it's waiting, like the anamnestic response in the adaptive immune system, it's waiting to respond. And whether it's a gene mutation and dysmetabolism or a viral infection leading to this lysosomal disruption, this priming is just waiting. And when it responds, it over-responds. And what happens is you get a systemic cytokine storm. And from that, you get an over-response of microglial cells in the brain, and you get delirium, and you can actually get cell death in the brain without having an infection by the virus. So this is a very important potential mechanism that has to be addressed as another reason for treating this systemic inflammatory response. Now, this effect of COVID-2 on Gaucher disease and other lysosomal diseases really bothered us at the beginning, because we were set up, certainly in Gaucher disease and others, for just an amplification or synergistic interaction with the disease, with the Gaucher disease or the lysosomal storage disease itself. But with reports from Italy, New York, Boston, New Haven, there were just a few cases of COVID-19 in Gaucher disease type 1. One reported death in a woman with severe or morbid obesity, and one report of a major non-ventilatory dependent lung disease that then resolved, but was severe. Other hospital cases, there are some, but they're relatively few. This could possibly be based mechanistically on the antiviral replication effect of type 1 interferon in Gaucher disease that could be potentially there in other lysosomal disease. There's also a high observance of the stay-at-home lockdown policies and avoidance of hospitals, even to the point of not taking enzyme therapy. But Gaucher disease and other lysosomal diseases must be considered a comorbidity in COVID-19. But the anticipated greater seriousness of the disease, the viral disease in these patients, has not been observed so far. Hopefully that'll continue to be the case. There are little data from other lysosomal diseases, including Fabry, the mucopolysaccharidoses, Pompe disease, which have heart, lung, and other tissue involvement, which COVID-19 also involves. And SARS-CoV-2 involvement of the CNS, the heart, the blood vessels, and the coagulation system remain nearly unexplored, and particularly in children, where all these systems are affected in the lysosomal diseases. I'll just end by saying I thank all my collaborators that have helped me over the years with the projects in Gaucher disease, the hundreds of patients with Gaucher disease, the NIH-led consortium on COVID-2, where we address the issues related to the care, and you'll hear more about that from Heather Lau in the future. And if you want to claim credit for this, you go to this website and you can get CME credit for this. And now I will turn the podium over to Dr. Inanova for her presentation. Thank you.